Can everybody hear me okay?
Yeah.
Well, that's great. Well, thank you very much for the opportunity to come and share with you folks what we're doing right up the road here in Boston College on Chestnut Hill. This is our Gasson Hall. I'm going to share with you the strategies that we are taking to manage cancer. We know that because we now know that it is a mitochondrial metabolic disorder, not a genetic disorder. And I will be presenting evidence for you to evaluate for yourselves as to your opinion on this whole thing. So what I like to do always in these lectures that I give is provide an update on our attempt to manage cancer. And some people call it an epidemic, other people call it a lost war. Is it better?
Better.
Yeah.
Better. Okay. Just have to know that. Okay, so you can look at these numbers. Now don't forget, this is coming from the American Cancer Society down in Atlanta. What they do is they log the dead. Okay, their biggest accomplishment is telling us how many people are dying from cancer. So if you look at this as 2013 to 2025, and you can see a relentless progression of new cases, deaths per year, and you can see deaths per day. You just divide the number in a year by 365, and you get an approximate number of folks that are dying in our country every day, which is about 1,700 people, okay? And if you divide that by 24 hours, you get about 70 people an hour are dying from cancer. And the increase is relentless, all right? So, you know, one of the strange things that always puzzles me is that we have all of these societies for raising money for cancer, the federal government included. Every year we're excited about how much new money we get from NCI, National Cancer Institute, Susan Komen, breast cancer, Stand Up to Cancer, Let's Run, Let's Climb. Everybody feels good about raising money for cancer, and what do we get? More cancer. Okay, how the hell do you explain that? What the hell is going on here? There's no accountability. You understand what I'm saying? There's no accountability. What we have done is brought cancer collection revenue, money for research as an end in itself, not a means to an end. We get excited, we raise more money this year than we did last year, everybody's back smiling, everybody's happy. But when we get more cancer, what's going on? So one of the ways we allow the public to know that we're making major advances is we say, and it says on the, if you read the papers from the National Cancer Institute, we have dropped cancer death rates by 33% since 1990. 1990 is when we instituted the anti-smoking campaign. So most of what we have achieved in managing cancer is a prevention strategy, not new drugs, a prevention strategy. And that sometimes is missed. People hear that and say, oh, we made tremendous, down here at Dana-Farber, all these places. Oh yeah, we made tremendous progress. No, you haven't. We have not made progress. So what can account for why we're not making any progress? And so the red line here is how many people would have died if they continued to smoke. So we've stopped cancer deaths from smoking because it's a prevention strategy. So we need to know what the origin of cancer is, so that when you understand the biology and biochemistry of the disorder, then you can make strategic approaches to managing the disorder. Cancer is dysregulated cell growth. That is what cancer is. People say, what is cancer? Cell division out of control. Caused by, is it caused by genetic mutations, like the world thinks? Or is it by chronic insufficient respiration coupled to compensatory fermentation, which may be a big blob of stuff, but that's what we do, okay? And I'm going to try my best to allow you to understand that, okay? Because some of these terminologies can be a little bit confusing at times. So It's a metabolic problem. What do you mean by that? So, but first of all, we have to say what the National Cancer Institute says cancer is. If you read on the top there, it says cancer is a genetic disease that is caused by changes to genes controlling the way cells function. And on the bottom over here, it says cancer is caused by certain changes to genes. And the industry, both academic and pharmaceutical, have been hunting for these genes that cause cancer now for about 50 years. And what we do is get more cancer. So there has to be something going on here. And it's become a dogma, a solidification of the mind. People go down on the streets here in Boston and say, what do you know about cancer? It's a genetic disease. Oh, well, how do you know? NCI says that. NCI can't be wrong. Oh my God. Confirmation biology supports all this. So people then go and do all this research and try to do this. So when you try to tell people something that's not true, and they've been solidified with dogmatic views, you get this kind of a response. They don't want to look at the data, they don't want to talk about it, and they don't want to hear about it. This is normal human behavior. This has happened historically. Now let's look at the data and see, do you think cancer is a genetic disease? That's the theory. It's called the somatic mutation theory. Theories drive approaches to management. If the theory is correct, you manage the disease. If the theory is incorrect, you don't often have success. We have some cancers that have no mutations, and they've been looked at. And the investigators that I have listed here, we've read these papers numerous times, I was saying, wow, they found a cancer that doesn't have any mutations. How is that linked to the somatic mutation, cancer gene theory? And guess what? They ignored too. I looked and I said, they didn't even discuss the cancers that had no mutations. I said, what the hell's going on with that? Then they say, well, not all mutations are bad. We have passenger mutations, and then we have the driver mutations. They're the real bad ones, and they're the ones we have to focus on. They're the ones we need to target with immunotherapies and all the things you hear about on TV. All right? Now we're learning that many of these normal cells in our body that we don't have cancer have loaded with mutations in driver genes. I say, how do you explain that? Yeah, it's a real head scratcher. Then we have some carcinogens that don't cause cancer, like asbestos. Cancer is extremely rare in Aboriginal tribes. In fact, the humanitarian physician Albert Schweitzer, who studied these African primitive tribes, was shocked. They don't have cancer. They do a lot of exercise, they eat a lot of meat. They don't have cancer. What's going on with that? The rarity of cancer in chimpanzees, our closest biological relative, 98% similar in gene and protein sequence. There's never been a documented case of breast cancer in a female chimpanzee. Breast cancer is one of the leading causes of cancer death in females in our society. The strongest evidence, normally I would go through, because I don't have enough time, and I would show you the evidence for each one of these things that I'm talking about. Deeper evidence, especially for the nuclear mitochondrial transfer experiments. What I can do here is simply show what we've produced as a summary of decades and decades of in vitro studies and in vivo studies. That means human, animals, and culture dish studies, coming up with this summary, overall summary. So you see the green cells, okay? Those are normal cells. Normal cells divide and they give, normal cells produce normal cells when they divide. The nucleus is healthy and the cytoplasm is healthy. The cytoplasm contains mitochondria. Mitochondria is what I'm going to be talking about. It's a mitochondrial metabolic disease. You're going to learn a lot about mitochondria. Mitochondria is responsible for our health. Mitochondria go bad, we have cancer, we have all these chronic diseases. Normal cells, we get normal cells. Tumor cells, the red, they have a lot of mutations in the nucleus, most of them for sure, not all, but most. And they also have abnormalities in the cytoplasm. The mitochondria in the cytoplasm. Abnormalities in the number, structure, and function of the organelle that provides life for us. And cancer cells beget other cancer cells, out of control, dysregulated growth. But what is causing this dysregulated growth, which is cancer? Is it the mutations in the nucleus? Or is it defects in the cytoplasm, in the mitochondria? Those experiments have been done over and over again, and I summarized all of those in my book and further studies. When you take the red nucleus out of the cancer cell with the supposed driver genes, and you put it into the normal green cytoplasm, you get cells that have regulated growth, not dysregulated growth. On the other hand, if you take the green nucleus from the healthy cell and put it in the cytoplasm of a tumor cell, you either get dead cells or cells that have dysregulated growth. This is the exact opposite of what you would have expected if cancer were driven by nuclear gene mutations, okay? These data, together with some cancer cells that have no mutations, driver gene mutations in normal tissues, aboriginal populations, you put all that together, and it's impossible to think that cancer is a genetic. You have to be cognitively impaired. So why the hell is the NIH telling us cancer is a genetic disease?
Because they want our money.
Well, I have different questions about that. Well, listen, there was a leader in the field of cancer genetics who looked at these data and said, if these data are true, the whole cancer industry has to be turned upside down. Therefore, the data cannot be true. That maintains the status quo. So if somatic mutations are not the origin of cancer, how do we get cancer? Well, my work was based on the work of Otto Warburg, the famous German scientist. Sam Apple wrote a nice book about him, an interesting character in the history of science. The most nominated person for Nobel Prize. He had one in 1931 for the discovery of the respiratory enzyme, but he had done some incredible work over the years. He said all cancers are a result from chronic insufficiency of cellular respiration, which is energy as we breathe. We breathe in oxygen, we get energy. Cancer cells have a defect in that capability. They get energy from lactic acid fermentation. This is not the kind of fermentation we just heard about. Fermentation is generating energy without oxygen, okay? All cells on the planet before oxygen came into the atmosphere 2.5 billion years ago were fermenters. There was no oxygen. They need oxygen for respiration. Cancer cells fall back on these ancient fermentation pathways. That was referred to the Warburg. Most people in the cancer field have heard the Warburg effect. It's the way the cancer cells generate energy without using much oxygen. They call it the Warburg effect, aerobic fermentation. You shouldn't be fermenting when oxygen is present. There was a lot of controversy about this for many years, many decades. But Warburg did not know, and what the field is now waking up to the fact that we have published here at Boston College. Cancer cells also use an amino acid. They ferment amino acids, especially glutamine. And glutamine is the most abundant amino acid in our body. So the cancer cells are actually using glucose and glutamine. These two fuels are driving the dysregulated growth. Enhanced fermentation due to mitochondrial dysfunction is the common pathophysiological malady in all cancers. So, people say, oh, I got this mutation, I got that mutation. It's all crap. Doesn't make any difference. The cell can't grow without a fermentation metabolism, and glucose and glutamine are the two fuels that are driving the dysregulated growth. Now let's look at the evidence to show that this is the case. As I said, the mitochondria drive the energy in our bodies. Mitochondria drive the energy. Without energy, we're all dead. We got to have energy. Okay, so this cell here with the stripes, those stripes are called cristae. You need to use an electron microscope to see these mitochondria in the cells. They contain the lipids and proteins of the electron transport chain, which allow us to get energy from the oxygen we breathe. The other, on the right side there, that's a mitochondria from a person who has glioblastoma, deadly brain cancer. You can see there's no stripes. Structure determines function. If the structure of the organelle is defective, the function of the organelle will be defective. That cell is not going to get energy the same way the normal cell will get it. It needs fermentation. But what about other cancers? Breast cancer, the same thing. You can see the nice stripes with healthy cristae in the breast cancer cell. But in the normal breast cell, but in the breast tumor cell, you see the vacuoles, you see the holes. Structure determines function. Those breast cells, those breast cancer cells are not going to be able to generate energy as efficiently as the normal cell. Colorectal cancer, same thing. Vacuole, these white holes there mean abnormalities in mitochondria. Structure determines function. If the structure is abnormal, the function will be abnormal, forcing those cells into a utilization of energy called fermentation energy, energy without oxygen. So what about all the other cancers? I went through it. This represents 95% or 97% of all cancer deaths. And we go back and look at the mitochondrial structure and function, and these cancers are all defective. If the mitochondria are defective in all these cancers, they have to get an alternative energy production, and that's through fermentation, energy without oxygen. I try to bring these just simple pictures here because I don't have time to go through the details. The glycolytic, this is normal, okay. We breathe oxygen, we breathe in air. Everybody's sitting here breathing, no zombies, right? I don't have zombies in here anymore. Have you ever asked where the zombie gets energy from? I ask my students that all the time. Well, my kids, yeah, that's really interesting. No, he's not breathing, he's like eating brains. Good brain. So anyway, you take the sugar, you metabolize it through the 10-step Embden-Meyerhoff-Pounds pathway, goes into the Krebs cycle for Hans-Krebs, and you break up, you generate new, what they call reducing equivalents. We breathe in oxygen, we produce tremendous amounts of energy, ATP. ATP is energy. And we blow out CO2, right? We exhale CO2 and water vapor. That's the waste product of normal respiration that we're all doing right now. You can see. And then we get a little bit of energy from these ancient substrate level phosphorylation pathways. Now look what happens in the tumor cell. You can just automatically see the tremendous shift from there to this. These are the ancient pathways of energy without oxygen. And the waste get much less energy from breathing, but the cancer cells are using non-energy sources called substrate level phosphorylation, and they blow out lactic acid and succinic acid. These are the waste products telling us that these tumor cells are using an altogether different source of energy than the normal cells. The cancer cells have been telling us this for decades, and the field has chosen to ignore that information. We didn't ignore it. Now, we put this in one of our new papers right here. This is a paper that just came out, ASN Neuro. You see, what is driving the dysregulated growth? Glucose and glutamine. They're driving the growth through the glycolytic pathway and the glutaminolysis pathway. Those two pathways are driving the dysregulated growth, blowing out, and there's synergy between these two pathways, allowing that cancer cell to synthesize all of the proteins and the lipids and the carbohydrates and everything that it needs by crossing those two pathways, blowing out the lactic acid and the succinic acid into the microenvironment, causing what we call acidification. You know, it's very interesting. If you go and look at the scientific literature in cancer, they say, oh, you know, those cancer cells are really hard to kill. They're tough, they're smart, they know this. You know, they protect themselves. That's why radiation doesn't work. Chemo, yeah, cancer cells are tough. But they're crazy. I mean, they're blowing out the two fuels that are creating acidification, making the therapy's not working. Yep. How do you stop that? You take away the glucose and glutamine. Is that complicated? Is that hard to understand? There is not a single trial anywhere in this country, in any major, even down the street here at Dana-Farber, MD Anderson, Sloan Kettering, the Moffitt, Hutch, doesn't make any difference where you are. Nobody's doing that. It's so simple to do that, and yet they don't do it. Because you know why? They don't think it's a genetic disease. Okay, so we'll just put this as our newest paper. Just came out in Bioenergetics and Biomembranes. Of course, you know, it's heavy, but we try to make it as open to everybody. How do you get cancer? Your mitochondria become damaged. How do you damage? How does the mitochondria become damaged? That's the oncogenic paradox, which we solved. Albert St. George said that in 1975. He said, geez, and I don't understand all these things that can cause cancer. How do they work through a common pathophysiological mechanism? Well, we went back. Carcinogens, even forever chemicals. Intermittent hypoxia, radiation can cause cancer in some people. Older ages, yes. Rare germline mutations, BRCA1, you hear about these mutations, that breast cancer and all these, they damage oxidative phosphorylation. Oncogenic viruses, chronic inflammation with microplastics, oncogene, all of these things will chronically damage the ability of that organelle to generate efficient energy. The organelle signals to the nucleus. It says, we're suffocating here. One of these things are blocking our ability to get good energy. The nucleus turns on what we call oncogenes. These are facilitators of bringing in the glucose and glutamine into the cell to replace the lost energy because the mitochondria are defective. Then you turn on the ancient pathways leading to dysregulated cell growth. Angiogenesis is the abnormal vascularization that you see in the tumors. Why? We have in our body, when cells go bad, we have a kill switch that kills the cell before it turns into a cancer. And that's controlled by the very organelle, mitochondria. When that's the kill switch, it goes bad, these cells bypass the kill switch, become dysregulated in their growth, cancer, blowing out lactic acid and succinic acid. I don't have time to talk about metastasis, which is spreading the cancer cells, but we know they're heavily glucose and glutamine dependent, so we know how to manage metastasis. The key here is good mitochondria, healthy metabolism. But chronic damage, over time, the cells begin to shift from one form of energy to another form of energy called substrate level phosphorylation, which is a fermentation energy without oxygen. The amount of that is directly linked to how aggressive your tumor is going to be, how hard it is, how malignant, they call the term the malignancy. So we know exactly what's going on metabolically in these cancer cells. This is the paper that just came out. It's called the Warburg hypothesis and the emergence of the mitochondrial metabolic theory. We have a new theory. When the theory is correct, we will drop the death rate significantly. It just takes time for the system to realize that. So if most cancer cells obtain energy through fermentation, what are we going to do to stop and manage the cancer? And that is, we're going to target glucose and glutamine. So one of the ways we do that is we can use diet, and we can use a variety of other things. So you can see, as you lower the glucose, you replace, cancer cells cannot use fatty acids or ketone bodies as energy because you need a good mitochondria to do that. If the mitochondria are dysfunctional and broken, you're not going to be able to transition to fatty acids and ketone bodies. You're locked into a fermentation with glucose and glutamine. Well, one of the ways you can do that is lower, yes, carnivore diets are great at doing that, no question about it. Water-only fasting, calorie restriction, or ketogenic diets. We realize now it's not just the ketogenic diet, it's the concept of nutritional ketosis. And carnivore diets bring you into nutritional ketosis, all right? The damn tumor cells suffer in nutritional ketosis because they can't use the ketones or the fatty acids. Let me show you the power of this. This is a mouse. It actually works better in humans. So this is a highly malignant brain tumor in the mouse. Ad libitum feeding is when he eats the high carbohydrate mouse chow diet, ad libitum, without restriction. You give the mouse the same diet, just restrict the calories. So 40% calorie restriction in a mouse is comparable to water-only fasting in humans. So people say, all I have to do is cut my calories back by 40%. No, you don't have to just drink water. So, no. I'll take the tuba thing. But you can see how dramatically reduced it. I was blown away by this. You know, I said, what the hell is going on? Because we worked a lot in epilepsy, so I knew how to work on these diets for years at Yale University. And then I said, wow, Otto Warburg said that a long time ago. I said, oh, who's this guy Warburg? Bring him back from the grave. You know. Now, this is an interesting study, the linear regression analysis. So each square is a mouse under a different diet. So, and then we measured blood glucose as the independent variable. As blood sugar goes down, beta-hydroxybutyrate, which is a ketone body, it rises. It's an evolutionary adaptation. This is what happens when you do a water fasting, carnivore diet. The ketone bodies go up. Okay, that's interesting because the cancer cell can't use that as a fuel. As blood sugar goes down, the size of the tumor and the malignancy goes down, right? The higher the sugar, the faster the tumor grows in the blood sugar. The lower the blood sugar, the slower the tumor grows. And when the blood sugar is down, you get reduced inflammation, reduced abnormal vascularization, and you start killing tumor cells. They're starving to death, right? So what do? Now, the clinical field went out and they started measuring blood sugar, breast cancer, brain cancer, all these different cancers. Wow, you have a lot of sugar. The tumor's going to grow up fast. You go down to Dana-Farber there, right? They got infused chemo in the arm and they're drinking Coca-Cola. What the hell is going on with that? Don't they look at the data? I mean, it's just so hard to understand, and it was repeated in humans over and over again. Somebody's not getting the word. What are you going to do? Write in crayon for crying out loud? So we developed a tool, glucose ketone index calculator, and this measures the ratio of glucose to ketones in the bloodstream. This is going to be the tool for managing all major cancers and chronic diseases. And we're working on a tremendous now big paper on this whole thing. And then we also developed the press pulse therapeutic strategy, which is a way to manage cancer. So you press therapies, which are ketones, you can do carnivore diets, get your GKI down 2.0 below, stress management, psychological stress, very important, physical exercise. And then we come in with the drugs, glucose targeting, glutamine targeting. You gotta be real careful, we're working this out. We take the stage 4 patient, we bring him into a state of management, okay, you get a management state. Then with improvements in doses, timing, and scheduling, we try to get long-term management. I never use the term cure. Cure is an arrogant term. Who knows whether you're cured or not? If you're living 4 or 5 times longer than you were supposed to be, isn't that good enough? What the hell's wrong with that? Okay, now I'm going to talk a little bit about human brain cancer, glioblastoma, terrible brain tumor. We killed Senator Edward Kennedy here in Massachusetts, John McCain, President Biden's son, Beau. So you know it's a deadly, they all went to the Mayo Clinic or someplace. But the problem is it's deadly. And here's the survival rate for glioblastoma. It doesn't look good. You know, it's so replicable, it's unbelievable. These are 5 institutions, they come tremendously the same, very poor survival. These are survival in months for this tumor. You know, the median survival is like 12 or 14, 15 months. You know, no improvement in 100 years. We have the Webb telescope circulating the Earth 1,000,000 miles, and we haven't made any advance in keeping glioblastoma patients alive. What's going on with that? I'll tell you what's going on with that. It's the way we treat them. What we do is we surgically debulk and then we irradiate the poor souls. And that frees up massive amounts of glucose and glutamine in the microenvironment, feeding the very cells you're trying to kill. And then they're wondering why the patients aren't surviving. Because you don't understand the biochemistry and biology of the disease you're working on. That's the problem. And we tried this on a guy from Egypt, a young man, Alexander, Egypt. My friend El Saka came in, we had this, he was about 27 years old, corn farmer. We fasted him. He had an awake craniotomy, removing the glioblastoma. We kept pushing off radiation as long as we could. We fasted him, we gave him keto-restricted diets. You must irradiate him. You must irradiate him. I said, what the hell, the guy's doing well. No, part of the standard of care. So he did survive the radiation pretty well, went back working in the farm, published a paper with 24-month follow-up, and then in about 30 months, the guy claims he has some headaches, and he dies. And El Saka did an autopsy on the guy's brain. They didn't find any tumor cells. He died from radiation-induced liquefaction of the brain. Can you believe it? I told him, what the hell are you irradiating? Oh no, you got to do it. I said, even in Alexandria, there's no flexibility. So we did another study in mice where we try to now target glucose and glutamine simultaneously using ketogenic metabolic therapy. So this is going to be the future. We do it first in mice to make sure we have success. So we bring the blood sugars down. We know we have to target the glutamine. You can't use diet to target glutamine. And you say the meat's got plenty. Don't worry. The meat is not enough to make the cancer cell grow with the glutamine, because the glutamine already in our blood is very high. So you need drugs to target the glutamine to bring it down. So we use this drug Don, 6-deoxynoreleucine, the most powerful drug that we know to block cancer is not being used, not allowed. It's a repurposed drug, you can't make any money on it. And they said it was too toxic. We figured a way to reduce the toxicity to make it work. Still not going to use it. So here's the animals that get the high carb diet. Boy, you want to die fast, you just eat high carbs. No question. Even the mice, just humans do it. This is the ketogenic diet. Yeah, they live a little bit better. The drug by itself, yeah, they do a little bit better, not spectacular. It's when you put the diet and the drug together, you get long-term survival, which is much, much better. So we know you have to target glucose and glutamine together to make this. And you push the ketones with the cancer cells can't use. Now, this is another tragic case. This is Brittany Maynard, a pretty young girl from California, diagnosed with glioblastoma in 2014. You can see here she's with her new husband. She gets a glioblastoma. She's treated with the standard of care. And you can already see her face is swollen. This is called moon face, high-dose steroids. And as I said, steroids increase blood sugar. So the treatments you're giving make the sugar go high and the tumor grow faster, okay? So now what she does is she writes a big article in People magazine, my decision to do suicide, assisted suicide, death with dignity. So she dies November, rather than, it's not good when your patients will kill themselves rather than take your therapy. That's the truth. I mean, these are the realities. Now, this is the guy who came to me in 2014 when we were very young at doing, we didn't really know. We had the concepts in our mind, but we didn't really work it out too well in people. And this guy, Pablo, glioblastoma, they said you have inoperable glioblastoma. And if you don't have radiation and chemo, you're going to be dead in 9 to 12 months. He said, well, the hell with that. I'm going to roll the dice, take my chance, because I want to have children. So no radiation, no chemo, no steroids. Oh, big brouhaha in England about this. He's from Devon, England. Oh no, you shouldn't do this. This is bad. So anyway, Pablo lived a little over 10 years, and he had 2 children. His wife was expecting, probably had the 3rd already. But here, we kept him 5 years. This is measuring Pablo. I wrote this paper, still being treated. I want to make sure he lived at least 5 or 6 years, because that breaks the record. But he collected very accurate data of his blood sugar and ketones. We built the GKI, and we show how low it is, right? Most of us here, most people in the society are around 50. So this is keeping the cancer cell managed. And he was doing really, really well. Now, what happened, a week before he passed away, we had a long conversation, and I said, you're gonna outlive me, because he was happy, go lucky. Now we have Pablo recovering from 4th surgical debulking of his original once inoperable tumor. So metabolic therapy allowed his tumor to be operated on multiple times. Unfortunately, he had a cerebral hemorrhage of 12 hours. So he has his thumbs up. He was having a big conversation. We were getting excited to have a call with him, and his wife emailed us, and he passed away from a cerebral hemorrhage. But he lived 122 months, right? If you can get 2 years out of a GBM, 3 years at the most, you're going to do really well. And this is our most recent study out of Greece. So my colleagues here, my Greek colleagues, Athanasius, Stephen, Julio, so they gave standard of care to these brain cancer, glioblastoma patients, standard of care, radiation, chemo, and this other stuff. But they also put them on a very tight metabolic therapy. It was a calorie-restricted Mediterranean diet, loaded with sardines, salmon, avocado, olive oil, all of this kind of stuff. And of the 6 patients who did the Mediterranean diet, 4 of them got 3 years or longer, whereas of the 12 who didn't do it, only 1. So if this were a drug, everybody would go wild about it. Look at all the survival, go with the 3 years and all this stuff. But once they found out it was a metabolic therapy, we can't use that. Now this is our big paper that just came out, where you can see a lot of names on here. Tom Duryea is working in my lab, the first author. We collected people from all over the world who want to make a difference in cancer outcomes and have them join us in this new society. So we're going to train and educate healthcare providers, whether you're a nutritionist, physician assistant, nurse, doctors, whatever, know how to use metabolic therapy for managing cancer, not only cancer, chronic disease. I'll show you evidence for that. Now this is another tragic case. This little boy is Danny Sheehan from Marshfield, Massachusetts, down here on the South Shore. Danny was diagnosed with a pineoblastoma, which the tumor cells moved down your spinal cord. And poor little Danny, you can see the moon face from the high-dose steroids and the treatments that he received. Here he is on the end, a week before he passed away, with his favorite toy. This should not have to happen. This should not have to happen. This is tragedy, okay? So I got so pissed off, we set up a pediatric model in our lab that mimics exactly what the tumor that little Danny had. And I have movies here, so these guys are eating, these guys got restricted diets, and bendazole, the parasite medication, and Dawn. Look at these guys, they're healthy and happy. These guys they took the regular diet, they're all dying. So we know that we can stop that in those pediatric clinics. We know that. They won't do it. Okay? You got to do the preclinical studies first to make sure these things work conceptually. And when you know that, then you can go into the clinic and you can get results that are even better than what we see in these mice. Okay, this is another case. This woman from Ohio, she had breast cancer. You can see from PET scan, she's lit up like a Christmas tree. The cancer cells went into the brain, into the bones, the liver, the lung. She was given 1 month to survive. She went to our colleagues in Istanbul, Turkey. They're following our metabolic therapy, and her scans became very clean. She almost died on the plane ride from Cleveland, Ohio to Istanbul, Turkey. My colleagues had to rescue her from death because of the trauma of the chemo and radiation, all the stuff she took. Anyway, they brought her back. They brought her, we give low-dose chemo. We don't throw out all parts of standard of care. We use them strategically when we need to use them. And she's doing that. Now, we just got a picture from her just this year. She's doing fine. She was written off as dead, go and make a peace with God. You know, what the hell? She's in Hawaii now, eating pineapples. You know, so, you gotta give it a break. Now we did it for advanced lung cancer. We got advanced guys coming out. I don't say the guy got non-small cell lung cancer. What are you doing? You're still alive at 10 years. Yeah, I'm doing great. I wrote a puff piece for Nature on get a lot of guys with stage 4 prostate cancer doing really well. And also dogs. This is amazing. So, this woman came to me. She said, "Oh, my loving dog is some sort of a pit bull or thing. Oh, it has a big mast cell tumor on its nose. Here's the dog's nose down here, this big ass tumor right here. It's a mast cell tumor. Look at it, she took the dog, she gave it raw chicken, cut the calories down, fish oil, raw egg, and a radish as a treat every now and then. And look how fast the tumor shrunk. You know, they wanted radiation and chemo, and they said the dog may live only 6 months or something. The dog is completely managed, and when you look at you can see the face, see a big tumor here. And then October 2016, the dog died in 2019 from heart failure in the arms of the owner. That's even longer than what normal pit bulls live, much less ones that have tumor on their face. Autolytic cannibalism. And the vet said we can't use this because you can't give dogs raw meat. Those people understand anything. Once he shoot off the floor for Christ's sake, you worry about getting a clean piece of chicken. You know? I mean, when you think about who we are as a species, sometimes you have to wonder, what the hell is going on here? So I don't like the term stage 4, terminal. We're all terminal for crying out loud. But why should we have to accelerate someone's terminality? We don't. Now, I put this in as a slap to the field. For 1,800 years, our ancestors tried to figure out where the different planets were in the solar system. Using equants, difference, all these different mathematical. And it was Copernicus who said, hey, maybe if we put the sun in the center of the solar system and the Earth is simply another planet, maybe it's not so complicated. Right? So what I'm saying is when you put the mitochondria in the center of the problem, it becomes far less complicated, not the nucleus, which is driving the entire cancer industry with all their mutations and all this kind of stuff. So mitochondrial metabolic theory can explain better the origin and management of cancer than can the somatic mutation theory. Cancer is a mitochondrial metabolic disease. The press pulse strategy, which I outlined, can also be effective, is an effective non-toxic strategy for managing the broad range of cancers. And we're coming out with a big paper, not only cancer, all these chronic diseases, all based on one form or another of mitochondrial dysfunction. The reason why we are a sick society is because we have sick mitochondria. The Paleolithic man did not have sick mitochondria. He died from infections and injuries. He wasn't dying from type 2 diabetes, cancer, cardiovascular disease, dementia. All of these are the expression of sick mitochondria in our cells. We're going to come out with a new concept with the GKI, and we're going to be able to show how we can manage chronic diseases and cancer. They're all linked to mitochondrial dysfunction. You keep your mitochondria healthy, and you avoid significantly reduced risk for all kinds of cancers and chronic diseases. We're developing a new society, International Society of Metabolic Oncology, trying to train as many people as quickly to get into the clinics so we can implement this therapy and keep people healthier. And listen to this. All of the money that I get from my research is coming from philanthropy and private foundations. You know, there are people out there that want to get involved with this. The paradigm change is coming. I don't know how long it's going to take, but once it hits, we're going to drop these chronic conditions and cancer significantly and save us a lot of grief. The only thing that we're going to piss off is the Grim Reaper. Carnivore brings you into low GKI. We tested all that. So we know that. All we're going back is as who we are. I tell a lot of people this. We are still Paleolithic man.
Haven't changed.
Haven't changed. Our technology has changed. We are the same individuals we were 500,000 years ago, but we're in a new environment that's causing all these chronic diseases because we love sugar.
Yeah.
We love it. It's a powerful drug. I know that. I love a jelly donut. Shake while you eat it.
All right, so I know what I'm going to do if something comes back. Thank you, Dr. Seyfried.